Um, so today I'll be talking about in silico metabolomics. It's a new concept, and it's an approach to try and look at what we call the dark matter of the metabolome. And it's been mentioned, I think, perhaps once, maybe by Kai or others earlier today, which is essentially the material that we can't identify. And it's a term borrowed from cosmologists and astronomers about the matter in the universe that we can't see, but we know is there. So I know there's a number of people who are probably new to metabolomics, and I know this is one of the first of a small number of sessions that have been held at ISMB on metabolomics. So I wanted to sort of give a bit of a background about the connection between metabolomics and the other things that most people at ISMB talk about, which is proteomics and genomics. And so I've illustrated this with this pyramid. Shujiao had talked about sort of the inverted pyramid with the metabolome at the bottom. I put the genome at the bottom as sort of the base. Genes code for proteins, proteins manipulate metabolites. And as you climb up this pyramid, there are increasing levels of influence by both environment and physiology. The genome is almost immutable. It doesn't really change with environment. It doesn't really change from physiology. The genome of the heart tissue is identical to the genome of muscle tissue or of the stomach or the brain. But the metabolome in heart or muscle or brain is very, very different. Same sort of thing is it with environment. What we eat or drink or breathe significantly influences our metabolome. Hopefully it doesn't influence our genome. Otherwise we'd all be sort of mutant monsters. So the metabolome kind of sits at that interface between the environment and the genome. So it makes it a really good tool for measuring the phenotype. So in terms of metabolomics, again, many people in the audience are probably familiar with it, but others may be new to it. The concept in metabolomics is somewhat similar to genomics and proteomics. We start with samples, tissues. They could be plants, microbial, or animal tissues. We extract them, not unlike what we would do with proteomics or genomics. And then in the end, we'll often work with some kind of biofluid, which is a little easier to look at. So rather than putting it through a sequencer, we tend to put those extracts into mass specs, which, like we do in proteomics, we'll also put them into NMR spectrometers or other things called gas chromatography mass specs, or sometimes we'll use infrared spectroscopy. These are the tools that are typically done to do chemical analysis, small molecules, as opposed to protein or DNA analysis. Now, the approach to doing analysis of biofluids goes back almost 100 years. The instruments of NMR and mass spec have been around for 50 or 60 years. The revolution, as I think Jeff and Shujiao highlighted, has been in the last 10 or 15 years, which has been the development of data analytic tools that allow us to look at, rather than just one compound at a time, to look at hundreds or thousands of compounds at a time, to look at really complex mixtures and read out the chemical composition. So the data analysis is where a lot of the focus is, and this is why bioinformatics and cheminformatics play a crucial role in metabolomics today. At least in mass spectrometry, the standard way of identifying compounds is to collect a GC or LCMS chromatogram, either a total ion chromatogram or extracted ion chromatogram, select a peak, in some cases in real time or in later time, look at the fragmentation patterns under those peaks. And typically a single peak will have 2, 3, or even a dozen different compounds under it. The only way you can identify those unique compounds is by separating them by mass to charge or retention time, or their fragment ion spectra. And what we're seeing here is some fragment ion spectra for the different molecules hidden under that peak, and we compare them to databases. And again, people have talked about the database comparison problem of where we may have a mass and we may have a MS, and we may even have retention time information. But that comparison requires databases. And the databases, as have been mentioned, are relatively small. They aren't as large as GenBank. They aren't as large as UniProt in terms of numbers of sequences. It's a few thousand to a few 10,000s of mass spectra. But by matching the mass spectra, what you observe to the actual ones in the database, just like sequence matching, you can identify, in this case, your compounds. Whereas in genomics and proteomics, we match to databases to identify proteins and genes. Now, metabolomics lags behind genomics and proteomics. We do not get complete coverage, whereas with genomics, it's standard to fully sequence all genes. With proteomics, we can typically identify about 10,000 proteins. With metabolomics, we're really happy if we can get maybe 150 to 300 compounds identified. There are two types of metabolomics. There's targeted metabolomics, and then there's untargeted metabolomics. The targeted approaches can use commercial kits. There are companies like Metabolon. There are also in-house assays that people develop. And typically with targeted metabolomics, people get anywhere from 150 to about 1,000 compounds that they can identify and semi-quantify. That's only about a tiny fraction, about 1 to 2% of the known metabolome. Untargeted metabolomic techniques identify many more features, up on the order of about 10,000 features. And through things like mass matching, typically they can identify about 700 to 1,000 compounds tentatively identified, and using more rigorous techniques that can maybe identify about 200 to 250 positive IDs. So whether you're looking at human biofluids, whether you're looking at cells and cell culture, water, soil for exposome studies, the same numbers keep on coming out, is that we're typically identifying between 1 and 5% of the peaks or the compounds that we know are there. So the coverage in metabolomics is still frustratingly low. Now, the reason for that is that metabolomics is inherently difficult. In the case of genomics, we're just sequencing 4 bases. The chemistry for base pairing and sequence characterization is well known. And for many years, fluorescence-based sequencing was based on chemistry. Proteomics, again, we're dealing with polymers that just consist of 20 amino acids. The chemistry, the cleavage, the possibilities and alternatives are also rather small when you only have 20 amino acids. In the world of chemicals, there's anywhere from 2 to 5 million biologically relevant chemicals. So the diversity, the chemical diversity that we have to deal with in metabolomics is hundreds to thousands of times more complex than it is for proteomics and genomics. And that's why metabolomics is difficult, and it's why where coverage is typically only 1 to 5%. There's also another challenge in that the tendency for many people is to turn to databases that are really not intended for metabolomics. So commonly preferred database is PubChem, which is a great resource, and ChemSpider, also a great resource. But these were designed to be chemical libraries of all known synthesized compounds that were ever assembled or written about. We've done some analyses, and of the compounds that are in these databases, less than 0.2% of them have actually ever left the lab. So that means that if they've never left the lab, they're not going to be found in you or an animal or a rat or a microbe or in the soil or in the environment. So that means that you can have up to a 99.8% false positive rate if you're trying to identify by mass matching to just a simple chemical database. The other point that's been raised is that getting chemical references, authentic chemical references, is both hard to do and expensive. If you go through standard sources and you have about $1 million, you can probably by about 3,000 meaningful chemicals, which then you can get and collect MS spectra. There are routes to get more, and certainly Metlin has been able to do that and has made a valuable contribution in that regard. However, overall, the experimental reference spectra, the things that we need in these spectral libraries, just like in sequence databases and structure databases for proteomics and genomics, they're very rare. They're just not a lot of them. And what's more is that even when we try and do our matches, even against super large databases like PubChem, most of the peaks that we see, most of the masses that we measure just don't match to anything. Nothing that's ever been synthesized, nothing that's ever been isolated, nothing that's ever been purified over the last 100 years. So that's another problem, is that most of those compounds, those unidentified compounds, are not in any database. And that's why we call it the dark matter. So the current thinking in many areas of the metabolomics community is that we have a pretty good handle on the known compounds, compounds that are synthesized, compounds that are in biochemical pathways, chemicals that are in natural products, chemicals in our foods, chemicals in cosmetics. There are ingredient lists. If you look at the side of a cereal box or any food container, the list of compounds is there. Human metabolism has been studied for 100 years. We have a pretty good idea of the main metabolites. But what happens to those starting compounds once they get inside our body, whether it's a food or cosmetic, a pollutant, or just the general metabolites that our body produces, they go through something called biotransformation, or phase 1, phase 2 metabolism, or they'll go through chemotransformation, which is just sort of spontaneous reactions. Some of them could be oxidation or reduction reactions. Some could be photo-induced reactions. Many of them are caused by the microflora in our body. Some are also changed due to our liver enzymes. So we start with a whole bunch of things that we know. Our body transforms them, and it produces this sort of green goop, which is a collection of mostly unknown compounds that are not in any database. So the current estimate right now is that the dark matter contains between 2 and 5 million compounds, none of which are in PubChem, none of which are in ChemSpider, none of which are in HMDB, none of which are in metabolites or any other database. So what can we do as a community? We can either try, as we did with the Human Genome Project, which is to gather a lot of money and a lot of resources, try and collect all the known chemicals, buy them, collect all the known xenobiotics, things that we found are known to be food additives in foods, all the drugs, all the pollutants, all the cosmetic, all the high production volume chemicals. And then we can then process them through artificial guts and artificial livers, or we can hire organic chemists to synthesize variations of them. And so we can get this collection of 2 million compounds by hook or by crook. And then we can collect their LCMS and GCMS and NMR spectra. And then we'd have our huge library, which is what we need. Now, minimally, if you were paying slave wages to people, you could get this whole thing done for about $2 billion. Probably a more realistic price would be about $20 billion. In these times, that's just not feasible. So it is unlikely that we will ever have the authentic compounds or the authentic spectra to characterize that dark matter. The other approach is to try and do it computationally. And I would argue that that's more feasible, more reasonable, and it's also cheaper. And so that's what we've been doing. And we're not alone. There are others, obviously, that are starting to do this. So the concept for in silico metabolomics is outlined here. The first task is to try and collect all of the known compounds, things that we know, are absolutely sure, are in either human systems or model organism systems or in the environment. I'll explain a little later, but right now we have a collection of about 330,000 compounds that we're very confident can be identified and are confidently have provenance in terms of where they are in the human body. The next thing is to try and then do this biotransformation computationally, to run it through tools that predict the chemistry of phase 1 and phase 2 metabolism, to predict microbial metabolism, predict promiscuous enzyme metabolism, predict spontaneous chemical reactions, all of which take place in organisms. And to predict those, and this is a tool I'll talk about called Biotransformer. And once we've been able to predict all of these biologically feasible structures, these things that are not in any database, the next step is to try and run it through some of the tools, some of which you've heard about. But our focus has been on a tool called CFMID, which is to predict MS-MS spectra. It also predicts GCMS spectra. We're working on tools to predict the NMR and the collisional cross-section. So we take these in silico structures and we create in silico spectra. And then with these referential spectra, we can start looking at our observed experimental data and do those database matches. So that's the concept behind in silico metabolomics. It's to create those libraries using bioinformatics and cheminformatics. Over the last 10 or 15 years, we've been working pretty doggedly at creating databases of known compounds. The Human Metabolome Database is an example, or HMDB, Drug Bank, collection of drug compounds, organism-specific databases like the yeast metabolome or the E. coli metabolome, food databases and phytochemical databases and toxic exposure databases. I'll explain these a little bit. The Human Metabolome Database, something that Jeff mentioned, the version 1 came out in 2008, I guess. And at that time, we had about 5,000 compounds. That's grown. We now know at least 114,000 compounds in the human metabolome. The database itself has lots of compounds. It has lots of pathways, lots of MS reference spectra, GCMS, NMR spectra. The database is useful because it not only is sort of referential, but it contains lots of information about the enzymes that process things, the receptors and transporters, the SNPs and reactions, links to diseases, referential values in blood and urine for biomarker analysis. Lots of descriptions on their concentrations, lots of tools for performing searches and queries, and it's integrated into tools like MetaboAnalyst. We've developed other databases to help expand our knowledge of the microbiome, E. coli being one, yeast being others, Pseudomonas, another microbe that we've been building databases for. And we're in the process of generating several thousand microbial databases like the ECMDB. But the current one has about 3,000 E. coli metabolites. These are linked to many reactions and genes and pathways, as well as a referential spectra. Drug Bank is another database we've been working on to try and collect both known small molecule drugs, experimental drugs, again, links to their targets and enzymes, along with the referential spectra. So in a human being, you may find a person with a few drugs. If you look at a population, you'll typically find hundreds, if not thousands, of different drugs in their system. And so Drug Bank also has become very useful in the field of both exposomics and metabolomics. Humans have to eat other metabolomes. And so in our effort to understand more about the human metabolome and more about the xenobiotics that show up in our body, we've been compiling constituents in foods, mostly raw or lightly prepared foods, things like coffee or wine or beer, as well as fruits and vegetables. Right now, the database has more than 70,000 compounds and are covering almost 800 different foods. And it's structured very much like HMDB and Drug Bank and ECMDB. In terms of trying to understand more about the toxic exposome or exposure compounds, we've been collecting data on pesticides and herbicides, solvents, pollutants, other things that show up generally at much lower levels, but are still findable and detectable in the body. So this database has tracked about 3,600 toxic compounds and also provides links to their protein targets and mechanisms of action. We've been extending some of this more to identify authentic biomarkers in a database called Exposome Explorer. It's looking at both diet markers and pollutant biomarkers and lots of detail on their concentrations and thresholds. So this is a joint effort with the International Agency for Research on Cancer in Lyon, which is where the ISMB will be next year. And this collaboration has also led to the identification of many compounds that are very useful for tracking exposures. The other thing that we see as mass spec becomes more sensitive is contaminants. And these are not necessarily toxic, but these are just things that we find in detergent, in clothing, in rugs and carpets, in cosmetics and perfumes, in paints and surfactants. And right now we have about 55,000. We're hoping to get around 80,000 by the end of the summer. And these are compounds that just typically aren't in most databases, but they tend to show up in your mass spectra when you are collecting things on biosamples. So I've given you a quick rundown on some of the databases that we've been collecting and archiving and updating. And together, there's about 330,000 compounds that we can provide provenance or source data that we are quite certain exist in either humans or in the environment that humans live in, or in most model organisms. So if we've got this set of known compounds, what can we predict? And this goes back to this pathway I was showing where we had known compounds from many different databases, their sources, source organisms. But the next step is predicting what happens to them once they hit the body, once they go into your stomach or into a rat's stomach or into a fish's stomach, what happens to them? And typically they get biotransformed. So biotransformation is the fate for most molecules. This is an example of a common drug, diazepam, and how it is biotransformed. In the liver, it's converted into multiple different metabolites, whereas in the gut, it's opened up and several other metabolites are also produced. And these are microbial metabolites. So the top set are phase 1 metabolites, usually mediated by cytochrome P450s or glucuronidases or different other phase 1, phase 2 enzymes. Now, there's a variety of tools to predict biotransformations, but almost all of them are commercial, and they actually have very tight restrictions on what you're allowed to do with those. So because of that, we developed our own tool, an open source, open access tool called Biotransformer. And this was an effort of Yannick, who developed Classifier, which Kai had mentioned. Yannick also developed the Biotransformer tool. It calculates the phase 1 and phase 2 biotransformations, gut transformations, environmental microbial transformations, promiscuous enzyme transformations. It has both a knowledge database, it uses some machine learning as well. That collection of both heuristic and machine learned tools allows it to do quite well. When we compared it to many commercial tools, it seemed to perform about 40% better and often was more accurate. This is an example of a compound which you'll typically find in things like apples and onions. It's called quercetin. It's a polyphenol. And when you eat an apple or have onion rings, it gets transformed. It'll be transformed to a wide range of different compounds, some of which are shown here. Eventually, most polyphenols are converted to something called hippuric acid. which we don't show here, but one of the precursors for it is also shown. So from one compound, it's possible to get up to 20 or more different metabolites. Some of them are known. Some of the ones that I've illustrated here have not previously been detected. We evaluated biotransformer and we're looking at it for a bunch of different compounds. We looked at drugs, we looked at food compounds, we looked at pesticides, a range of other compounds, both primary and secondary metabolites. And we compared them to a commercial program called Meteor. And we looked at things like precision and recall and a number of false positives and true positives. And generally, we found that the precision and recall for biotransformer was about 10 to 20% better than the commercial one. Now, the commercial tools are generally very limited. They only perform phase 1 metabolism. So biotransformer is able to do the other types of metabolism as well, phase 2, microbial, environmental, and promiscuous. We've converted biotransformer to a web service, and it allows you to predict sort of in a limited way different reactions and products. Depending on how much you ask it to do, it can take anywhere from 5 to 30 seconds. We've been running in the background biotransformer for the last few months to generate from those known compounds that we have in our databases to the unknown or dark matter. And right now, we've generated about 2.1 million compounds, and we put them into a database called the Biotransformer Database, or BTDB. It's very much in a beta version, so don't expect a lot. But we wanted to make that available for some of our colleagues and for the community. What this has is now about 2.1 million compounds, 90%, 99% of which are not in any database, all of which are biologically feasible, all of which have some provenance where we can either identify a specific enzyme or pathway or process that should lead to that kind of metabolite. We have information on the structure, the names, the formula, the mass. Of course, we'd have very little information about what these things are. They are really hypotheses, but the intent is that this now represents a likely collection of molecules for what we would call the dark matter. Generating this database took thousands of CPU hours. We're regenerating it because we've improved biotransformer quite recently by using more machine learning techniques, and those have boosted its performance by another 20% in terms of sensitivity and specificity. We've added more complex biotransformation steps. We've corrected some errors. And then, of course, the database itself, we will try and improve it so it's a little more user-friendly and include all of the predicted MS spectra, retention indices, and others in that resource. We've been testing it because, again, it was sort of a hypothesis, but we wanted to see if it would work in real life. We worked with collaborators in France where they looked at how epicatechin, which is a T metabolite, would be biotransformed in rats and mice, and they collected urine samples. And using biotransformer, they were able to identify 22 known compounds, but that took about 3 months of literature search to verify those. And then the suggested 12 other novel metabolites, which they're trying to confirm now. So literally in about a 30-second run on biotransformer, they were able to get something close to 35 metabolites identified, which subsequently required months to validate. But the point is that biotransformer offers this opportunity to identify many unknown or hard to find compounds where they just simply don't exist in reference libraries. So biotransformer generates compounds and hypothetical structures, but what we measure in metabolomics is not a compound structure. We measure a spectrum. We measure MS spectra or GCMS spectra or NMR spectra or collisional cross-sectional area. So the idea is to take these synthetic in silico compounds and generate observables. in silico. And so this is what CFMID is about. That stands for Competitive Fragment Modeling and Identification. It was developed by a graduate student who worked in my lab, Felicity Allen, and she published this about 5 years ago. We converted it to a web server, and it allows people to take a compound and predict the spectrum. It allows you to take a spectrum and annotate an MS spectrum, all of the fragments, but also allows you to take an MS spectrum of an unknown compound and to predict what the structure is. It's designed specifically for QTOF instruments. We're adapting it to work better for Orbitrap instruments. It uses machine learning techniques, similar to the probabilistic graphical models or hidden Markov models that PIMA uses. And we found that as you train it more, it does better, which is the nice thing about machine learning. When it first released, it did quite well in a number of tests, and we've been improving it steadily over the last few years. We've added more deep neural net tools into the fragment modeling. We've expanded the training set. We've improved both the chemical feature and topological representation to help with the fragmentation. There were problems in the early versions with ring cleavage. We've improved that. And then we've added rule-based schema for compounds where machine learning doesn't do so well. So for lipids, acylcarnitines, acylcholines, polyphenols, and a few other classes. So with those improvements, we've been able to enhance the performance of CFMID for lipid spectral prediction. On the right is an example of a predicted and experimental spectrum, blue and red. It's called a mirror diagram. So the stuff on blue on pointing up is the real spectrum. The stuff pointing down on red is the predicted spectrum. And we measure performance by what's called a Jacquard score, the number of peaks that match. And you can see it's not perfect, but in terms of the peak positions, it did quite well. We're comparing it to another tool called Lipid Blast, which predicts lipid spectra. And using machine learning or rule-based methods, we can see a distinct improvement. We've also developed other improvements with things like polyphenols and multi-aromatic structures. On the left side is the old version of CFMID, and we're measuring which is equivalent to the Jacquard score. It's called a DICE score. A perfect score is 1. So the old version, you're getting scores of 0.25 and 3.5. With the new version, matches and the scores are up around 0.6 to 1. We've compared CFMID over the years. We have a version 2, a version 3, and a version 4. And these are the Jacquard scores, or equivalent to DICE scores. And we're looking at different collision energies. So these are MS spectra. So the early version, CFMID 2.0, did really poorly in lipids, moderately okay for the DICE score in terms of electron volt collision energies, low, medium, and high. The newest version, version 4, which is hopefully going to be coming out this summer, some people are already using it, is performing about 20 to 30% better than the earlier versions. The rule-based methods also substantially improves the lipids. And then we've been using what's called the CASME data set, which was a collection of about 200 compounds. And that's sort of a nice benchmark to assess performance. And we can see that each time we've improved CFMID, the number of compounds that we could confidently identify increased. So right now, CFMID is able to identify about 80% of the compounds from that CASME data set. And this is, using a dummy set of about, I think it was 40,000 compounds that the compounds could be. So it seems to work quite well, and it's getting progressively better. And this suggests that in silico metabolomics is something that could really help with the compound identification in that dark matter problem. We're not the only people doing this. People are making use of CFMID. They're making use of biotransformer. And there are several publications that have appeared over the last few years where people have integrated the tools I've just mentioned in pipelines to do metabolite or natural product identification, and to create a pipeline that makes the novel compound identification quite efficient and quite fast. The one that was described on the right, they were able to identify an average of one completely novel compound every 2 days, whereas typically it takes 2 to 3 months if you work really hard, and in some cases 2 to 3 years if you're like the rest of us to identify and characterize a novel compound. So using in silico metabolomics, whether it was the case for analyzing T metabolites or identifying novel compounds in plant constructs or identifying natural products, really seems to work. So to wrap up, I just wanted to highlight a couple points. Typically, compound identification of unknown compounds is really, really hard. And we, as a community, tend to identify the known compounds, which only represents about 1 to 5% of the features. We're in a tough situation in the metabolomics community. We don't have sufficient MS spectra or compound libraries now or even in the foreseeable future. So the only route, I think, for now to get around that problem is to do in silico techniques, to come up with better or larger spectral libraries to create feasible biological compounds using what we know about chemistry and biochemistry. And that by using these predicted compound libraries, these predicted MS spectra, these predicted NMR spectra, predicted collisional cross spectra, we can actually perform identifications. I think these tools are improving year by year. I think it's exciting to see what both machine learning and careful chemical inference helps and does to this. And I think I also wanted to emphasize that this is not a pipe dream. People are already doing this. They're using it. They're putting them into pipelines, and they are working. So with that, I'd like to thank my collaborators and the funders. I'd also like to thank Jeff Shaw, who was my former PhD student, who's been doing fantastic work in developing his own sets of tools for metabolomics. And I also like to thank the organizers and thank all of you for sticking with this and listening to the presentation. Thanks.
Great, thank you very much, David. So it's very enlightening. There are a lot of questions in both Q&A box and in the panelist box. Let me start with the one in the Q&A box. So it's the biotransformer based only in human enzyme transformation, or do you include microbial functions, etc.?
So the microbial transformation includes many, many microbial groups or sets. So these would be things that you would find in the gut. It also includes microbial transformations that occur in soil as well, but in some respects, those also align with what we find in the gut. So it's quite diverse. And we are trying to work or add some of the cytochrome P450 transformations that we would typically see in plants, and some of the glycosylation processes that are also seen in plants. But that'll be a different version of biotransformer.
So Cecilia Noika is asking, are you planning to expand FoodDB to include more cooked and processed food? I guess she doesn't like raw food.
Yeah, so it's a very difficult thing to do. There is data already at the USDA for many of the processed foods for limited numbers of compounds. Our view is that we wanted to try and capture most cooked and processed foods start with raw foods. And so there are some transformations that happen with cooking. And obviously, if you make, lasagna, you've got tomatoes and pasta, and there's many other ingredients. In principle, you would have all of the components in FoodDB for the recipe for lasagna. So you could assemble that from the recipe, the likely constituents for lasagna. And so that's our perspective on trying to include prepared foods. The cooking is a difficult thing to predict what's happened with them, and to collect the data is really, really difficult.
So there are a couple questions on the same theme. What's the differences between the biotransform database versus other compound ID databases and the database from Oregon National Lab and other tools like Magma, etc.?
So the biotransformer database is a collection of predicted compounds, all of which are biologically feasible. Probably none of them are compounds you will find in databases like PubChem. You won't find them in the Argonne Lab. You won't find them in any other database. So they are unique, distinct, and biologically feasible. And not necessarily all correct. So they're essentially tools to test or assess. And they all have some provenance, meaning that they tell you which was the starting compound and what were the enzymes or pathways that led to it. So it's not just sort of a random collection of machine learned chemical structure generator. It's one where it's actually computed the likelihood and enzymatic provenance of those compounds. The database itself still doesn't have referential spectra. That takes many, many CPU months of calculation. So we're running that now. And as I said, version 2 of the database will have that referential data. So the idea is that you would use it similar to the human metabolome database or METLAN, except it would be a collection of compounds that is all in silico.
Lorenzo Calvello wants to know if you can use predicted by transformation to search for compounds that are likely to interact with protein complexes and nucleotide acids.
So people are already starting to use some of these biotransformed compounds to start screening, doing QSAR work, ligand docking work. Again, that might help people suggest bioactivities for certain compounds. Certainly people working in the field of natural products and foods are really interested in this because most food metabolites or micronutrients are heavily transformed. And although many of you hear about the polyphenols and the antioxidant activity, most people in the field of polyphenol research now realize that the health effects from polyphenols come from the biotransformed metabolites, not from the polyphenols themselves.
So there is a question that really deserves people's sympathy. So the question is, I have metabolites from Metabolome Inc. without any IDs. So how can I use them if I do not have any identification or pathways? I don't know how that come about. I guess the person received some data from a commercial vendor and some of the data do not come with identifiers.
Yeah, so Metabolon typically will provide a list of compounds that it formally identified, and then it'll have compounds which it sees consistently, but it doesn't have formal identifications on them. You can also, I think, if you get the really cheap Metabolon analysis, I guess they just give you the list of identifiers, but no metabolites. So the trick is, pay them more money and they'll give you more information. The alternative is if you have the mass to charge information that they've released, then it is possible to search through various databases to see if you can get a match. But at least if you're purchasing or acquiring data from Metabolon, the only way you could get more information is to pay them more money.
Another question is that I'm still confused. What is the input for your algorithm to identify novel metabolites?
So we take the structures of our known compounds from our databases like HMDB and DrugBank, and Exposome, Explorer, and FoodDB. So there's about 330,000 structures. These are in mole files or SDF files or SMILES. And we feed those mole files or SDF files, those electronic files, into Biotransformer. Biotransformer looks at the structures, makes predictions on which bonds of metabolism, sites of metabolism will happen, where the metabolic reactions will occur. It runs through hundreds of different possibilities. It has a knowledge base that it looks through. It has machine learning tools. And from there, it generates a whole bunch of, again, mole files, SMILE strings. In this case, about 2.1 million. So it's electronic data that goes in and electronic data that comes out, and they are structures, electronic structures.
Well, the last question here is tricky. Did you ask for NIH funding to identify metabolites?
So we are part of an NIH Common Fund project, working with Tom Metz and PNNL, that's focused on exactly this, in silico metabolomics. And they are working actively in the area of collisional cross-section prediction with a tool called DarkChem. And my group is working actively in the mass spectral prediction. And the idea is to merge both the CCS predictions with MS predictions to help identify novel metabolites using this in silico approach.
Let me add to that. Actually, I put the information on the Metabolomics Consortium from NIH in my talk. So the link is metabolomics.info. Actually, NIH recognizes that metabolite identification is the most urgent task in the field. So they actually funded 5 centers working on compound identification. So at metabolomics.info, you can find information on the projects. People can also find other educational resources and funding opportunities.